Okay, so this is an elderly paper from 2004 by a Japanese group, obviously. Changes in oral microbial profiles after periodontal treatment as determined by molecular analysis of 16S RNA genes from the Journal of Medical Microbiology. And so what they're looking at is how do the organisms on your teeth differ before and after going to the dentist. And it's a little bit worse than that. And so they picked 3 people with periodontal disease. They take before samples, both saliva samples and tooth scraping, subgingival samples. And then they treat them by a process called scaling and planing. I don't want to know what that means. Hopefully it involves Novocaine. They clean the teeth both above and below the gums. They clean it right down to the dentin or whatever. And then they give them lessons about oral hygiene. And then 3 months later, they come back and test their teeth and their saliva again to see what difference it makes. They do some real-time PCR, but that's just basically confirmation. It's basically the tRFLP they use. This was an early paper, and so they also, at the end, go back and do some ribosomal sequencing just to confirm the results. They weren't very impressive, but I'll show you those in a minute. They did terminal RFLP. They used a 6-forward labeled primer, 27 forward, and 1492 reverse, and they digest with HHA1 and MSP1. Ideally, they would use a third enzyme to confirm every one of their data points, but they don't do that. And so how do you interpret these? These are the patterns they get. This particular one is the, which one is it? HHA1. And so there are 3 patients, A, B, and C. There are periodontal samples and saliva samples, and there's before and after. What do you see? There's some pretty significant before and after. I mean, certainly there's variation from one person to another, right? That's pretty obvious. There's some differences between the periodontal and saliva, but they're not so great. The before and after, in some cases, they're really dramatic differences, right? Here. You don't even have to know what those peaks are to quantitate that, right? You could scan those things in, measure similarity and difference, and create a tree or some other measure of similarity to show, to quantitate how much difference there is before and after. Is that satisfying? Maybe a little, right? Wouldn't you like to know who's there? So they do exactly the kind of analysis we talked about. They do 2 digests, one with HHA and one with MSP1, and then they go through and deconvolute the patterns to identify the identity of the organisms represented by all those peaks. And for the most part, they do a pretty good job. There are peaks in all these where they can't identify who it is, but again, they find the standard sort of organisms in a large part that you would expect to find in your mouth. Both the good ones, things like Streptococcus salivarius. Here's Strep mutans, the cause of dental caries, right? Fusobacterium is a good one. Porphyromonas gingivalis is probably not good. And so you can go through these things and see that, if you're careful, you'll see that the before and after Porphyromonas and Prevotella essentially go away. So that's a good thing, right? Whereas, let's see, Flavobacterium, I don't know what that one is. Strep mutans comes back. That's actually good. Strep mutans kind of gets a bad rap. It does cause cavities, but it's a normal part of your tooth biofilm. It's just that if you eat a lot of sugar, it causes problems. Here's Peptostreptococcus. No doubt that's not good in your gingivas, right? This is an anaerobe fermenter, proteolytic organism, certainly going to cause lots of inflammation. And the good news is it went away after treatment. This is the same pattern then with the different restriction digest. I won't go through that. Oh, I should point out one thing. The before and after was particularly effective for 2 of the patients, and one of the patients went back essentially, I think that's patient A, went back right to where it was before. Any idea why that might be? What's that? Behavior? Yeah, right? And you guys have been brushing your teeth and flossing and all that kind of stuff your whole life, right? Is someone giving you a lesson that's going to change that? Probably not. The 2 of them did a pretty good job, though. I'll point out, by the way, that these are extremes. I mean, these people clearly have very serious gingivitis. When this paper was done, this tRFLP technology was new and not the sort of thing that people were going to take at face value. And so they did, they did one thing. They did real-time PCR for a very specific reason, which is that they don't see any treponema or any spirochetes at all in any of their tRFLP patterns. And yet, if you go in and look under a microscope, you can see they're there. Can any of you think of a reason why this might be? They aren't in the RFLP library. That would be one reason why they wouldn't be there. It turns out they actually are. Yeah?
The restriction enzymes.
Yeah, so if the restriction enzymes don't have any sites in there, you get the full-length thing and you get no information. That's one reason. Another reason? What if they're, go ahead. Yeah, the primers. What if the primers don't hit it? Let me give you an example. You don't see any lactic acid bacteria in here because 1492 reverse doesn't work on lactic acid bacteria. And so that's the kind of thing that people say. The other thing is that when you do this kind of analysis, you can only see the most abundant members of the population. All these little peaks, if it represents a couple of % of the population, that's really obvious when you're looking at spirochetes under a microscope. But it's not really obvious when you're looking at a complex banding pattern. And so there's a variety of reasons why they didn't see them. And so they go back and do real-time PCR to quantitate some of these organisms in there to verify at least the basic pattern of their results, which they do. The other thing they do to verify the tRFLP is they go through and they make banks of sequences. And so they do ribosomal, they take the ribosomal PCR, sequence a bunch of them. They only did 90 clones before and 88 clones after. So we're not really looking at a very statistically relevant sampling here. But you do see kind of a pattern, even if it's informal. And so, for example, you see that organisms like Eubacterium go away. Eubacterium is basically Clostridium without endospore form. Probably not the sort of thing you want in your teeth, right? Fusobacterium is unchanged. Where's another one? Neisseria pharyngis, this is a normal flora organism, is more abundant after treatment. Peptostreptococcus essentially goes away. That's good. Porphyromonas goes away. Prevotella goes away, except for this one. Who else? A bunch of streptococci are more abundant after treatment than before. Streptococcus is a good normal part of the biofilm community. And Veillonella is more abundant after treatment than before. And so basically, there's evidence here that you are transforming that microbial community from one type to another, probably in a good way. And certainly when you look at these people's teeth, they're in better shape afterwards than before. And the paper describes that for the 2 out of the 3 patients. An interesting question, I think, about this paper, and one that I've asked on the exam in the past. If you were going to do this experiment today, this is 10 years old, how would you change it? what would you do if you were doing this experiment today? Yeah.
Well, you would change your primers.
You would change your primers. Would you use tRFLP at all? You could. I think you could also make the argument that high throughput sequencing would be a more appropriate approach. Yeah.
I don't know if it's worth asking, but I would change some of the methodology, like use a larger sample size.
Larger sample size is a big one. This is 3 guys, all right? The statistics on this are going to stink no matter what you do. And so you're going to have to use a larger population. You're going to have a more diverse population. You don't want to know just about one males in one ethnic group, right? You're going to want more diversity than that. What other things might you change?
If you did ribosomal sequencing, you could increase the number of clones you had in each.
Right. So instead of looking at 180 sequences, you could be looking at 180,000 sequences, and then you can do statistics in some way. What else might you do?
I want to survey too the habits that they like figure out exactly what people were doing with the internet time.
Yeah. So what's your diet? How did your dental hygiene change before and after? I would argue that 2 time points doesn't cut it, right? Wouldn't you want to have a couple of time points before to set a baseline, to see how much variation there is from time to time, and then a series of time points afterwards? Maybe it takes a while for things to settle down. Maybe after 3 months, everything looks good, and after 6 months, they've just gone back to the same old thing as before. What else would you might want to change?
You said that they pick people that have periodontal disease.
Yeah.
Why would they pick people that have that? Like, did they say at the beginning why?
They did, and for the very mundane reason that you're not going to subject a person to scaling and planing unless you have a reason to do so. Nevertheless, if you get volunteers, it might be useful to take a look at that, right? What about people with something other than periodontal disease? What about people with dental caries? What about people with halitosis? What about, the list goes on and on, right?
What about people with healthy teeth?
Yeah, with totally healthy teeth to start with. Might it be different for incisors than molars? What about people with mercury fillings, like me? Hopefully you guys don't have mercury amount of fillings anymore. If there's one thing I would change about my history, well, I wouldn't do that, but one thing I would change is having amalgam fillings. It's a terrible thing to do, having a mouthful of mercury. The only thing worse is getting them taken out, which gives you a giant dose of mercury all at once. But what about people with fillings? Because people with gingivitis are likely to have fillings, right? Does that have an effect on this? I'll bet you it does. Whenever you read one of these papers, I think it's important to ask yourself, what would you have done differently? In this case, it's easy because it's an old paper. But even for new papers, it's important to ask yourself, what could they have done differently to make this better? Not as a way to second guess the people, because they probably did the best they could under whatever their circumstances were. But given infinite resources, are there ways in which you could do an experiment that would make the point better in some way? All right, good. We're done with this. I think next time